Hyphens in paper titles harm citation counts and journal impact factors. According to the latest research results, the presence of simple hyphens in the titles of academic papers adversely affects the citation statistics, regardless of the quality of the articles. The phenomenon applies to all major subject areas. Thus, citation counts and journal impact factors, commonly used for professorial evaluations in universities worldwide, are unreliable. This breakthrough finding poses a fundamental challenge to the rule of the game in determining the contributions of papers, journals, and professors. It is unveiled in a paper titled Metamorphic Robustness Testing, Exposing Hidden Defects in Citation Statistics and Journal Impact Factors by Ji-Kwon Zhou, Th Zhe, and Matt Witheridge, recently published in IEEE Transactions on Software Engineering, the top journal in the field. Th Zhe is an honorary professor in computer science at the University of Hong Kong HKU. Ji-Kwon Zhou received the PhD degree from HKU and is currently an associate professor in software engineering at the University of Wollongong, Australia. Matt Witheridge is a PhD student at the University of Wollongong. Scopus and Web of Science are the two leading citation indexing systems. Scopus provides the citation statistics to support the Times Higher Education World University Rankings and the QS World University Rankings. Web of Science provides the journal impact factor that supports the ranking of major journals. Because of the importance of these two indexing systems, it is essential to assure their quality. In particular, robustness testing refers to the verification of the system's ability to deal with erroneous inputs or unexpected situations. For example, can the indexing system handle a citation properly if there is a minor typo when quoting the paper title? Professor Zhe and team members proposed an innovative method named Metamorphic Robustness Testing to verify Scopus and Web of Science. The in-depth study uncovered robustness defects in both systems that might produce erroneous citation counts for papers with hyphens in the titles, so that the journal impact factors subsequently computed are problematic. Back in 2015, Lechford and colleagues conducted a large-scale study on Scopus, and found that papers with shorter titles tended to be cited more than those with longer titles. Their results were widely reported in international media including Science and Nature. On the contrary, Professor Zhe and the present team find that it is actually the number of hyphens in the title that serves as the more dominating factor for citation counts. Usually, the number of hyphens is correlated to a paper's title length, thus giving the misinterpretation that citation counts depend on title length. Citation practices vary across subject areas. Publications in certain fields may have systematically higher citation counts than in other fields. For example, one may argue that papers in chemistry, where paper titles often carry hyphens as part of the chemical nomenclature, only receive relatively limited numbers of citations, giving rise to a spurious negative correlation between hyphens and citations. Hence, the team carried out focused studies on journals in specific subject areas. The results indicated that hyphens adversely affect the citation counts of papers even if the study is only limited to some particular discipline. To build on the findings at the article and discipline levels, the team investigated the impact of hyphens in paper titles at the journal level. Journal Impact Factor (JIF) is a common metric for determining the citation frequency of an academic journal. It is frequently used to represent the relative importance of a journal within its field. A software engineering field-wide study reveals that the higher JIF-ranked journals are publishing a lower percentage of papers with hyphenated titles. The team further conducted an analysis of the validity of the research to avoid falling into the trap of equating correlation with causation. Our results question the common belief by the academia, governments, and funding bodies that citation counts are a reliable measure of the contributions and significance of papers. In fact, they can be distorted simply by the presence of hyphens in article titles, which has no bearing on the quality of research. Similarly, our results also challenge the validity of journal impact factors, said Professor Zhe. These surprising results are of interest not only to professors seeking tenure or promotion, but also to the senior management such as presidents, deans, and heads. They are applicable to all faculties in any university, he added. Physicists teleport logic operation between separated ions. Physicists at the National Institute of Standards and Technology (NIST) have teleported a computer circuit instruction known as a quantum logic operation between two separated ions, electrically charged atoms, showcasing how quantum computer programs could carry out tasks in future large-scale quantum networks. Quantum teleportation transfers data from one quantum system, such as an ion, to another, such as a second ion, even if the two are completely isolated from each other, like two books in the basements of separate buildings. In this real-life form of teleportation, only quantum information, not matter, is transported, as opposed to the Star Trek version of beaming entire human beings from, say, a spaceship to a planet. Teleportation of quantum data has been demonstrated previously with ions and a variety of other quantum systems. But the new work is the first to teleport a complete quantum logic operation using ions, a leading candidate for the architecture of future quantum computers. The experiments are described in the May 31st issue of Science. We verified that our logic operation works on all input states of 2 quantum bits with 85 to 87% probability, far from perfect, but it is a start, NIST physicist Dietrich Liebfried said. A full-scale quantum computer, if one can be built, could solve certain problems that are currently intractable. NIST has contributed to global research efforts to harness quantum behavior for practical technologies, including efforts to build quantum computers. For quantum computers to perform as hoped, they will probably need millions of quantum bits, or qubits, as well as ways to conduct operations between qubits distributed across large-scale machines and networks. Teleportation of logic operations is one way to do that without direct quantum mechanical connections. Physical connections for the exchange of classical information will still be needed. The NIST team teleported a quantum-controlled NOT (CNOT) logic operation, or logic gate, between 2 beryllium-ion qubits located more than 340 micrometers millionths of a meter apart in separate zones of an ion trap, a distance that rules out any substantial direct interaction. A CNOT operation flips the second qubit from 0 to 1, or vice versa, only if the first qubit is 1, nothing happens if the first qubit is 0. In typical quantum fashion, both qubits can be in superpositions in which they have values of both 1 and 0 at the same time. The NIST teleportation process relies on entanglement, which links the quantum properties of particles even when they are separated. A messenger pair of entangled magnesium ions is used to transfer information between the beryllium ions. See infographic. The NIST team found that its teleported CNOT process entangled the 2 magnesium ions, a crucial early step, with a 95% success rate, while the full logic operation succeeded 85% to 87% of the time. Gate teleportation allows us to perform a quantum logic gate between 2 ions that are spatially separated and may have never interacted before, Liebfried said. The trick is that they each have 1 ion of another entangled pair by their side, and this entanglement resource, distributed ahead of the gate, allows us to do a quantum trick that has no classical counterpart. The entangled messenger pairs could be produced in a dedicated part of the computer and shipped separately to qubits that need to be connected with a logic gate but are in remote locations, Liebfried added. The NIST work also integrated into a single experiment, for the first time, several operations that will be essential for building large-scale quantum computers based on ions, including control of different types of ions, ion transport, and entangling operations on selected subsets of the system. To verify that they performed a CNOT gate, the researchers prepared the first qubit in 16 different combinations of input states and then measured the outputs on the second qubit. This produced a generalized quantum truth table showing the process worked. In addition to generating a truth table, the researchers checked the consistency of the data over extended run times to help identify error sources in the experimental setup. This technique is expected to be an important tool in characterizing quantum information processes in future experiments. This work was supported by the Office of the Director of National Intelligence, the Intelligence Advanced Research Projects Activity and the Office of Naval Research, pain-free, thanks to evolution. Frickin' mole rats are insensitive to many different kinds of pain. As an international research team led by the MDC's Gary Lewin reports in Science, this characteristic has even allowed mole rats to populate new habitats. Thanks to a genetic change, the Highveld mole rat is able to live alongside venomous ants with painful stings that other mole rats avoid. Around 10 years ago, naked mole rats at the Max Delbruck Center for Molecular Medicine (MDC) became famous almost overnight. That was thanks to the work of Gary Lewin from Berlin and Thomas Park from Chicago, who together investigated the strange sensory world of the naked mole rat. Park and Lewin showed that these African burrowing rodents were remarkably resistant to pain. In a 2008 article in the journal PLOS Biology, they reported that the mole rats felt no pain when exposed to acid or capsaicin, the substance that gives chili peppers their heat. The experiments on the mole rats drew global attention. For this latest study, just published in Science, Lewin teamed up with colleagues in South Africa and Tanzania to further test more instances of pain insensitivity. The insights provided by our studies of these animals should help, among other things, in the development of new pain-relieving drugs, says Lewin, who leads the Molecular Physiology of Somatic Sensation group at the MDC. Working closely with a world expert on mole rat biology, Nigel Bennett at the University of Pretoria in South Africa, Gary Lewin, Thomas Park and colleagues explored how naked mole rats and 8 other related species respond to 3 substances that usually cause a brief burning sensation on the skin of humans and other mammals. Those substances were diluted hydrochloric acid, capsaicin, and allyl isothiocyanate, AITC. AITC is what gives wasabi, the popular condiment served with sushi, its extremely hot taste. The idea was that mole rats are naturally exposed to these and similar substances in the wild. Only the Highveld mole rat is impervious to wasabi's sting. In the Science article, whose lead authors are Ole Eigenbrod and Carlian Debus, the scientists report that 3 mole rat species proved to be insensitive to the acid. Interestingly, those 3 species are not particularly closely related through evolution. 2 species did not show evidence of pain after having a capsaicin solution injected into their paw. Others briefly lifted their paw, or licked at it, which shows us that those animals did feel a brief sensation of pain, explains Lewin. Only a single mole rat species proved to be impervious to AITC. This was not the naked mole rat, but another burrowing rodent called the Highveld mole rat, which is named after the region in eastern South Africa where the animals are exclusively found. This was a thrilling finding for us, says Lewin. AITC attacks amino acids in the body and can thus destroy proteins. That is why all the other species we know avoid coming into contact with the substance. The Highveld mole rat was the only species in the experiment that had no problem with AITC. It's all down to altered ion channels. In order to uncover the molecular reasons for the mole rat's remarkable resistance to pain, the researchers took sensory tissue from the spinal cord and dorsal root ganglia of all 9 species studied. In the dorsal root ganglia there are clusters of neurons that transmit pain signals to the spinal cord. With the help of state-of-the-art sequencing technology, we were able to compare the activity of around 7,000 genes inside that tissue, reports Lewin. Relatively quickly, the team observed that the activity of 2 genes was altered within the animals that felt no pain. These genes contain the blueprint for the ion channels TRPA1 and NaV1.7. It is already known that these 2 channels are involved in the perception of pain. AITC and many other irritants found in roots, one of the mole rat's main food sources, activate TRPA1, explains Lewin. That is why, over the course of evolution, many species have downregulated the gene for this channel. But the wasabi channel is the only one to be completely switched off in the Highveld mole rat, Lewin says. He and his team went on to discover that this is due to a particularly active gene for another channel, the constitutively open channel NALCN, known as a leak channel. The research group found that the expression of this channel was the only one that was significantly altered in the Highveld mole rat. Mole rat pain resistance can be switched off. Lewin was particularly surprised by the results of a further experiment. When we blocked the NALCN channel by administering a drug that blocks the channel, the Highveld mole rat suddenly became sensitive to AITC after all, he reports. But just one day after the antagonist had been administered, the animals regained their indifference to the substance. From the thousands of genes we were looking at, we had obviously found the very gene responsible for the Hyphens mole rat's remarkable pain resistance, says Lewin with a smile, delighted that luck was so clearly on his side. Daniel Hart, a PhD student working with Nigel Bennett, discovered that Hyphens mole rats often share their burrows with Myrmecaria natalensis, commonly known as the natal droptail ant. These insects are known for their aggressive nature and highly pungent venom, says Lewin. When this ant venom was injected into their paw, all mole rat species in the study experienced a brief moment of pain, all except the Hyphens mole rat. But when the researchers blocked the NALCN channel in the Hyphens mole rat, these animals became sensitive to the venom. New findings could help pharmaceutical research. Lewin surmises that throughout the course of evolution, the Hyphens mole rat has clearly acquired a highly active gene for a single ion channel, which has allowed it to make its home in places that are avoided by other species of mole rat. The MDC scientist regards this as another wonderful example of how environment shapes evolution over the long term. On the more practical side, this discovery could well lead to the development of highly effective analgesics, he adds. The Hyphens mole rat has showed us that high expression of the NALCN channel seems to be a very effective way of alleviating pain. Earth recycles ocean floor into diamonds. The diamond on your finger is most likely made of recycled seabed cooked deep in the earth. Traces of salt trapped in many diamonds show the stones are formed from ancient seabeds that became buried deep beneath the Earth's crust, according to new research led by Macquarie University geoscientists in Sydney, Australia. Most diamonds found at the Earth's surface are formed this way, others are created by crystallization of melts deep in the mantle. In experiments recreating the extreme pressures and temperatures found 200 kilometers underground, Dr. Michael Forster, Professor Stephen Foley, Dr. Olivier Allard, and colleagues at Goethe Universität and Johannes Gutenberg Universität in Germany, have demonstrated that seawater in sediment from the bottom of the ocean reacts in the right way to produce the balance of salts found in diamond. The study, published in Science Advances, settles a long-standing question about the formation of diamonds. There was a theory that the salts trapped inside diamonds came from marine seawater, but couldn't be tested, says lead author Michael. Our research showed that they came from marine sediment. Diamonds are crystals of carbon that form beneath the Earth's crust in very old parts of the mantle. They are brought to the surface in volcanic eruptions of a special kind of magma called kimberlite. While gem diamonds are usually made of pure carbon, so-called fibrous diamonds, which are cloudy and less appealing to jewelers, often include small traces of sodium, potassium and other minerals that reveal information about the environment where they formed. These fibrous diamonds are commonly ground down and used in technical applications like drill bits. Fibrous diamonds grow more quickly than gem diamonds, which means they trap tiny samples of fluids around them while they form. We knew that some sort of salty fluid must be around while the diamonds are growing, and now we have confirmed that marine sediment fits the bill, says Michael. For this process to occur, a large slab of sea floor would have to slip down to a depth of more than 200 kilometers below the surface quite rapidly, in a process known as subduction in which one tectonic plate slides beneath another. The rapid descent is required because the sediment must be compressed to more than 4 gigapascals, 40,000 times atmospheric pressure, before it begins to melt in the temperatures of more than 800 degrees Celsius found in the ancient mantle. To test the idea, team members at the Johannes Gutenberg Universität Mainz and Goethe Universität Frankfurt in Germany carried out a series of high-pressure, high-temperature experiments. They placed marine sediment samples in a vessel with a rock called peridotite that is the most common kind of rock found in the part of the mantle where diamonds form. Then they turned up the pressure and the heat, giving the samples time to react with one another in conditions like those found at different places in the mantle. At pressures between 4 and 6 gigapascals and temperatures between 800 degrees Celsius and 1100 degrees Celsius, corresponding to depths of between 120 and 180 kilometers below the surface, they found salts formed with a balance of sodium and potassium that closely matches the small traces found in diamonds. We demonstrated that the processes that lead to diamond growth are driven by the recycling of oceanic sediments in subduction zones, says Michael. The products of our experiments also resulted in the formation of minerals that are necessary ingredients for the formation of kimberlite magmas, which transport diamonds to the Earth's surface.